Hey Corny, what's up?
Hi.
Do you know what designer babies, glowing animals, and rubber's grain has in common?
Oh, that sounds like the beginning of an awful joke. But no, I don't, so go ahead.
It's actually really interesting. It's CRISPR-Cas9.
Excuse me?
Wait, you don't know about CRISPR-Cas9? So you also haven't heard about all those crazy scientists which call themselves biohackers?
Biohacker? CRISPR-Cas9? That sounds like nerds in cereal. But now you've got me interested. Tell me.
Okay, let me start with the basics. So what is CRISPR-Cas9? Cas9 is an enzyme which is used in nature by bacteria as a viral defense. When a prokaryotic cell is attacked by a virus, a part of the viral DNA is inserted into the bacteria's own DNA between the repeating so-called CRISPR sequences. CRISPR is an acronym for clustered, regularly interspaced, short palindromic repeats. The CRISPR section is then translated into an RNA strand, the cRNA. The cRNA fuses together with the tracer RNA and the Cas9 enzyme to form a large molecular complex. If the same virus infects the cell again, the CRISPR-Cas9 enzyme recognizes the viral DNA at the PAM region, binds the viral DNA at the appropriate place, opens the DNA strand and cuts near the PAM region to disable the viral DNA. To use the CRISPR-Cas9 complex in eukaryotic cells, the tracer RNA is simply connected to the appropriate cRNA section, and then the crafting can begin.
Okay, and what can be done with it?
There are a lot of possibilities to apply this technique in molecular biology, plant and animal breeding, parasite control, and in human therapy. So it can be used in gene therapy to treat infectious diseases or cancer. In personalized medicine, there could be new approaches too. And also it is possible to create replacement for animal models or to enhance them. So first let me explain an example from human therapy. For example, there's a person who has beta thalassemia, which is an hereditary synthesis disorder of the beta chains of the protein portion globin in hemoglobin. And normally these patients require a stem cell donor, but often no donor can be found or such a transplantation is not feasible due to various risk factors. Then the patient depends on regular blood transfusions causing stress and potential life-shortening side effects. The solution could be CRISPR-Cas9. First, hematopoietic stem cells are taken from the patient. They are then processed in a laboratory by the CRISPR-Cas9 method. Before the edited cells can be reintroduced into the patient, the bone marrow is destroyed. Subsequently, the patient receives his or her gene-modified cells in a blood transfusion. These blood stem cells settle in the medullary cavity of the bone and begin to form functional blood cells. So all in all, an alternative hemoglobin gene is activated, enabling the manipulated cells to produce so-called fetal hemoglobin, which takes over oxygen transport. This approach was already successfully applied in a patient who is until now transfusion-free and has a normal blood value. So with further progress in research, the lives of many people with various diseases could be improved. And I have another very interesting example in crop sciences. Worldwide, agriculture is suffering from threats caused by abiotic and biotic stresses. With CRISPR-Cas, crop varieties may be improved, which leads to higher quality and yield rates. For example, wheat is often attacked by wheat mildew, which leads to yield losses. The method CRISPR-Cas9 is used to knock out genes which are causing susceptibility to powdery mildew in plants expressing this gene, either MLO or EDR1. This technique then improves powdery mildew tolerance, so that these plants have then an enhanced resistance to powdery mildew.
That sounds really promising.
Yeah, that's true. But despite all the possibilities, the conventional application of CRISPR has some limitations and downsides. A major hurdle is to decode and translate the respective genomic sequences. Also, CRISPR has a potential for off-target effects, which are effects that we cannot yet determine exactly. So we could cause devastating consequences if we use this technique without hesitation on humans and the environment. This is why the European Court of Justice is already setting up stricter regulations.
Sounds reasonable. And didn't you mention something about biohackers?
Yes, mostly in the USA there is a so-called biohacker community. These are people who have purchased CRISPR-Cas toolkits for about $170 to $700 for personal usage. Some think that the technology and knowledge should be available to the public. Others are just crazy and want to try out some gene editing on themselves. For example, to generate more muscle mass and thicker skin, which of course still have uncertain consequences.
Crazy. I'm curious how it will all develop, but thanks for explaining.
Yeah, you're welcome. Bye.
See you.